Hi everyone, and thank you for joining today. My name is Dungan, and I'm a postdoctoral research fellow at the University of Melbourne in Australia. I'm going to kick off today with some heavy facts. Climate change-induced thermal stress is the leading contributor to coral death. In the summer of 2015, there are more than 2 billion living corals on the Great Barrier Reef. Half of them are now dead. This image shows the beginning of that process, with healthy tissue on the left that is full of symbiotic algae, to bleached and aposymbiotic on the right, just a few days later as a result of thermal stress. Given this dramatic loss of coral from human-induced climate change, but the optimism for developing assisted evolution strategies to mitigate these effects, our lab has been exploring the possibility of bacterial probiotics to protect corals. Today, I'm going to talk to you about a strategy our group has used to identify potential probiotic candidate bacteria. This work was published last month in Microbial Biotechnology. To start with, I'll quickly walk you through the basic biology of cnidarians, why we should care about these organisms, and how climate change is impacting their survival. I'm going to start by introducing the main characters in this story. Cnidarians are largely marine animals and include corals, as shown here, hydras, jellyfish, and sea anemones, among others. Today I'm focusing on corals and sea anemones. These organisms are holobionts and would not survive without the relationships they form with other microbes. These microbes include an endosymbiotic algae of the family Symbiodiniaceae, bacteria and archaea, viruses, and fungi. It's important to note the Symbiodiniaceae transfer photosynthate to the surrounding coral cell. This fixed carbon can account for up to 95% of the coral's energetic needs. For this work, we use a model organism for corals, Exogenous diaphana. This sea anemone shares the same microbes that are present in the coral holobiont. When 50% of the reef dies, as it has in just the past few years, we're losing more than just biodiversity. The Great Barrier Reef has an economic value of $56 billion, which comes from fisheries, tourism, coastal protection, pharmaceuticals, and its value as an icon to Australia. This image shows a coral thicket from its healthy state here on the left to bleached, yet alive, and then dead and covered in algae. This dead reef is no longer providing the same ecological or economic services. The conditions that lead to bleaching, that is, prolonged periods of elevated sea surface temperature, are occurring more and more frequently. Isolated colonies or small patches of bleached coral are not necessarily cause for concern. However, mass bleaching events that span tens, hundreds, or even thousands of kilometers are a huge concern. We've had 3 mass bleaching events. I'll quickly go through a simplified version of what happens during the bleaching process. During thermal stress events, the symbiotic algae that reside inside the host incur damage to their photosynthetic machinery. This results in an accumulation of reactive oxygen species, or ROS, that further damage the photosynthetic machinery, and ROS compounds, such as hydrogen peroxide, may even leak out of the Symbiodiniaceae cell into the coral host cells. This results in an inefficient and reduced transfer of photosynthate, and the final step is a loss of the algal symbiont, thus revealing a white skeleton beneath a translucent layer of tissue. Hence, this phenomenon is called bleaching. Our hypothesis is that we can selectively add beneficial bacteria with the capability to neutralize reactive oxygen species, therefore maintaining a stable host-symbiont relationship and preventing coral bleaching. Now, we just need to figure out which cnidarian associated bacteria might be able to neutralize ROS. As precedent for our hypothesis, probiotics are routinely used in agriculture to enhance nutrient uptake and drought resistance, and in aquaculture for disease prevention and to reduce nutrient buildup. In Exogenous, the administration of probiotics mitigated the effects of the whitepox pathogen. Probiotics have been used in corals as well, with administration of potentially beneficial bacteria enhancing coral survival during oil exposure and reducing the effects of bleaching during a simulated thermal stress event. The mechanism of the latter is, however, unclear. Thus, our aim was to develop a consortium of Exogenous-sourced bacteria strains capable of scavenging extracellular ROS. Free radical scavenging was used as a proxy to quantify the antioxidant properties of host-associated bacteria. The first step in designing a potentially beneficial bacterial consortium is building a repository of bacteria. To establish a culture collection, replicates of 4 anemone genotypes were homogenized, diluted, and spread plate onto marine and R2 agar. 842 bacterial isolates were purified, sequenced, and stored in our culture collection. Isolate identity was determined from partial 16S rRNA gene sequences. This neighbor joining tree gives an overview of the phylogenetic relationship between the 842 Exogenous-associated bacterial isolates inferred using partial 16S rRNA gene sequences. These isolates covered 6 phyla, indicated by shading over the tree, with proteobacteria split into the classes gamma proteobacteria, which is highlighted in light blue, and alpha proteobacteria in dark blue. Importantly, this diversity of cultured Exogenous source bacteria is similar to what has been reported from metabarcoding data for other Exogenous strains, as well as corals, with the key exception that no Endocercomonas, a common coral-associated bacteria, was found. Free radical scavenging properties for each strain were measured using the stable free radical DPPH, which is reduced in the presence of an antioxidant molecule, undergoing a color change from violet to yellow. Of the 842 isolates, 709 were qualitatively screened for their ability to scavenge free radicals, divided into positive, weakly positive, and negative. After application of a DPPH solution, bacteria that could scavenge extracellular free radicals formed a halo around the colony, as shown on the left, which was absent in the negative strains, as indicated on the right. The difference between positive and weakly positive designation was based on the time it took for a halo to form, where positive individuals took less than a minute and weakly positive individuals took 1 to 3 minutes. From those isolates, 98 strains representing 8 families and 18 genera were then quantitatively assessed for free radical scavenging. This method involved creating an extract of the bacterial cell-free supernames, combining in a 1-to-1 solution with the DPPH reagent, and measuring free radical scavenging as the percent change in absorbance at 517 nanometers compared to a DPPH. The first thing I want to do is look at the results for one family, Rhodobacteriaceae. On the x-axis, you have the percent free radical scavenging, which is measured as the percent change in absorbance at 517 nanometers, and the y-axis has each bacterial strain. For these assays, I used a series of ascorbic acid standards. These are on each of the next few figures. From here, I could select the high free radical scavenging strain to be used in the bacterial consortium. One point of criticism for other probiotic work has been the absence of controls. In the coral probiotic experiment that I mentioned earlier, for example, the addition of bacteria may have simply served as an additional food for the corals, and their beneficial effect may have had nothing to do with the trait the bacteria were selected by. Thus, we decided to establish a consortium comprised of conspecific or congeneric bacteria with a low free radical scavenging ability to act as a negative control. From Eltermonadaceae, 3 conspecific pairs of high and low free radical scavenging strains were selected. Similarly, 1 high-low free radical scavenging pair from Microbiology and Flavobacteriaceae were selected for inclusion in the consortiums. In total, we identified 6 conspecific pairs of isolates with a high and low free radical scavenging ability belonging to the families Eltermonadaceae, Rhodobacteriaceae, Flavobacteriaceae, and Microbiology. Selected high free radical scavenging strains, those that are on the right of the figure, were those that had a mean DPPH free radical scavenging greater than 50%, low standard error between replicates of a given strain, and consistent growth in R2A broth media. The conspecific low free radical scavenging strain averaged less than 50% free radical scavenging and was not significantly different to the media negative control, which is shown here in gray. When we map these high free radical scavenging, and those are the blue arrows, and low free radical scavenging, which is the orange arrows, back to the original neighbor joining tree, we can see that we have a diverse set of potential probiotic candidates. Our aim was to develop a consortium of Exogenous-associated bacterial strains capable of scavenging extracellular ROS using a high free radical scavenging as a proxy. And we did that by establishing a high free radical consortium and, as an experimental control, creating a low free radical scavenging consortium comprised of conspecific or congeneric strains. Our next step was to see if we could identify a genomic basis for these different phenotypic responses. Comparative genomics between the high and low free radical scavenging strains was uninformative, as there were either too many or too few SNP differences in the core genome, and accessory genome differences were difficult to interpret. We then focused on pathways that could produce metabolites with antioxidant properties, which then be correlated with our free radical scavenging test and could be useful when it comes to mitigating bleaching. Several pathways are included in the manuscript, but today I want to talk about a more recent finding that is not included in the manuscript, and that's mannitol biosynthesis. Mannitol biosynthesis is something that we're really excited about. Mannitol is a carbohydrate, specifically a sugar alcohol, and an antioxidant, making its potential biosynthesis by bacteria relevant to our free radical scavenging assay. Cnidarians exposed to elevated temperatures in the presence of mannitol have reduced rates of symbiodiniaceae loss, photosynthesis and respiration rates indistinguishable from their pre-temperature stress rates, decreased rates of DNA damage, and prolonged survival. The cumulative result from these studies suggests that increased biologically available mannitol could potentially mitigate bleaching and prolong the life of corals during periods. Mannitol biosynthesis in bacteria has two main pathways. First, fructose 6-phosphate can be reduced to mannitol 1-phosphate with the enzyme mannitol 1-phosphate dehydrogenase, or MTLD. Mannitol 1-phosphate is dephosphorylated to mannitol by mannitol phosphatase, and that gene is HXPA. Bacteria can also catalyze mannose 6-phosphate to fructose 6-phosphate via MAN-A. Alternatively, mannitol is directly synthesized from fructose by mannitol 2 dehydrogenase, MTLK, without the synthesis of mannitol 1-phosphate. Now let's see how our candidate bacteria fare when it comes to these genes. We found 11 of our probiotic candidates, all but the Marinobacter high free radical scavenging strain, to contain mannitol biosynthesis genes. Specifically, the low free radical scavenging Marinobacter strain had HXPA only. Micrococcus and Winogradskiella strains had the HXPA and MAN-A genes, while Labrenzia and all 4 Altromonas strains had MTLK genes. This is the first time that mannitol biosynthesis genes have been reported in cnidarian-associated bacteria. And this is really fascinating since we have the full spectrum of genes in these candidates. There is the potential for mannitol biosynthesis, and this should be explored further. Is there a future for mannitol producing bacteria and climate change resilience? Well, there's strong evidence that exogenous mannitol addition can rescue corals from bleaching. And I have shown here that cnidarian-associated bacteria have the potential to synthesize mannitol. Interestingly, bacterial MTLD has been transformed into higher plants. The transgenic plants were able to produce and accumulate mannitol. To continue on this path, We need to quantify mannitol production of coral-associated bacteria under different growth conditions. Consider transforming mannitol biosynthesis genes to key bacteria taxa, such as the dominant bacterial symbiont Endozochomonas, and understand that life exists in context. Bacteria in culture just do not have the same environmental conditions as those living on or inside of a coral host. Studies should push to assess bacterial functions in hospitae. Now let's think back to the main aim of developing a consortium of bacteria to scavenge extracellular broth. We can consider what impact a consortium would have for the future of corals as the climate continues to change. While we tend to bring up the risks and unintended consequences of bioremediation strategies, such as probiotics, we absolutely have to consider the known consequences of no intervention at all. In this case, if no interventions are taken to protect corals from climate change, they will all be dead by the end of the century. Instead of inaction, my lab proposes the application of potentially beneficial bacteria as a way to mitigate climate change. These bacteria are sourced from corals, easy to grow, and accumulate biomass, and represent a sustainable way to remediate the effects of climate change in corals. With more research, there is the possibility of using maternal transmission of bacteria to coral larvae as a more sustainable option for probiotics. However, I would be remiss not to mention the challenge that lie ahead of us. We need to test the free radical scavenging consortiums that I mentioned today on cnidarians during a heat stress experiment. Stay tuned for this manuscript to hopefully be available by the end of the year. It is also unknown whether introduced microbial communities can remain associated with corals and influence fitness traits over extended periods or into the next generation of corals. The cellular processes that cause bleaching are nuanced and complex. While I have spent this talk discussing reactive oxygen species, there is evidence that nitrification, reactive nitrogen species, and other agents can lead to bleaching as well. Thus, we need to keep pursuing other biological features that might mitigate bleaching, such as denitrification, alternative carbon fixation, and the production of antimicrobials. Getting the bacterial consortium into the ocean is a challenge as well. Current approaches to make probiotic application in a marine environment feasible include bioencapsulation of probiotic members with feed, concentrating bacteria using weighed-down enclosures, slow-release beads, probiotic paste, and drone dosing at the onset of a thermal stress event. However, the feasibility of getting permits for these studies is very low, as we do not know what unintended consequences there may be on the coral reef. Ultimately, we must move away from simply documenting the demise of coral reefs to exploring sustainable interventions that can increase corals' resilience to climate change. Without such interventions, such as probiotics, coral reefs simply will not survive. Funding and team that supported this research, I look forward to answering your questions and discussing other ways that bacteria might be used to protect corals from climate change.